Very nice presentation. I'd like your comments relating homozygous lines and probably your insights on the production of secondary metabolites are so important. Over the last 30 years, we have been trying to do it. I'd like your comment on that, because you have got a lot of experience on gene expression, please.
Well, in our lab, we also are very interested in secondary metabolites. And in fact, we have been working with a MEP pathway, isoprenoid pathway, that produces carotenoids, that also produces Taxol and many of these. Of course, understanding differentiation of the chloroplast and understanding the regulation of the pathway is essential. So we need to understand the regulation of the pathway. Right now, for example, we are doing transplantomics, and those for you that are not very familiar with the term, it means that we are transforming chloroplasts. And we are transforming chloroplasts because these organelles have a beauty in the fact that are transmitted by maternal tissue. So we avoid cross-contamination of transgenics, that is a problem, but also we do not have silencing in chloroplasts. And in some cases, we can produce 50% of the protein of the plant when we transform chloroplasts. However, again, we need to understand the basic biology. We are overexpressing one of the limiting enzymes of the pathway, which we know that is very important. However, the protein has their own wheels because it's regulated in the folding. And we now realize that we have to understand much more profoundly this enzyme. So I think it's potential, and I think it's very important. And I think it has to be the production and try to manipulate these organisms, and I think it's very promising. But we need to know basic science too.
Thank you. Luis, please.
Yeah, okay. Very nice talk, Patty. If you have these signals coming from the chloroplast that act in the nucleus, do we know anything about the receptors in the nucleus that are targeted by these signals?
I wish I have. But let me tell you one thing. ABA, which is also an apocarotenoid, actually binds to transcription factors that have a domain called the START domain. And a START domain is a lipophilic domain that can bind apocarotenoids. And this is also true even in some animals. So what we speculate, but we have not been able to demonstrate, is that maybe these apocarotenoids have the capacity to leave the plastid, the chloroplast, the plastid, and then bind to specific transcription factors. We are, of course, trying to see which are the transcription factors that are upregulated, and also see which ones regulate those transcription factors, and see if any of them have a specific domain that might be a hint or clue about what could be. But that's an idea. We do not have a specific regulator, well, a specific binding to or receptor. But yeah, that's more or less what we think.
Okay, Carlos?
Yes. I'm curious, is it known what are the factors, the DNA polymerase and the RNA polymerase that transcribes or that replicate the DNA in the chloroplast and the mitochondria is encoded in the nucleus. So my question is it known whether these polymerases are synthesized and sent to the chloroplast, in this case, in a continuous manner, just at a basal level, or whether these polymerases are actually requested by the chloroplast when the chloroplast is going to divide, or where it is going to express more of the genes in their genome, in the case of RNA polymerase?
Yeah, it is. There are two well, actually there are three, but mainly two polymerases that are important for chloroplast to transcribe the genes in the chloroplast. The first polymerase is nuclear, but the second is chloroplastic. And the first polymerase, the nuclear, is a T7 type polymerase, and then it goes and starts the polymerase inside of the chloroplast, which then turns on several of the genes in the chloroplast. And the first polymerase, the nuclear polymerase, is regulated and is expressed mostly at the very early transition of the chloroplast development. And then it goes down. And then the polymerase from the chloroplast takes off. But they are, the polymerase, the nuclear polymerase required, and also the polymerase encoded in the chloroplast requires sigma factors. And the sigma factors are nuclear, and they are regulated at different moments of the plastid. And there is not well known when each of these sigma factors are regulated. But things are even more complex because you do not only regulate transcription. You know, transcription in the plastids, in the organelles, and that's true also in mitochondria, probably not the most important things to regulate. The two organelles appear to be regulated more than the post-transcriptional step. So you have editing, you have internal splicing, and all these things make it extremely complex how fast the transcripts stay or are degraded, and if they are translated or not. They have to be processed, otherwise they are not translated. And some of them, they have to be edited, otherwise they are not translated. So addition, translation, transcription are points of regulation, certainly. But we still do not know exactly at one moment what signals. For example, the light regulates the expression of the polymerase. And that's the first signal for chloroplast to start. But then there are multiple other regulations that come into play, and we believe that also the chloroplast tells it, we need more, we don't need it. So I'm not sure if I answer all your questions.
Make one more commentary just quickly. Some years ago, we tried a crazy experiment with mitochondria, not chloroplast, but I assume that there are similarities. The idea was to try to actually start putting genes back into the mitochondria that had actually been sent to the nucleus throughout during the endosymbiosis. And we were able to put the gene of the RNA polymerase, the nuclear RNA polymerase, into the mitochondria and integrate it stably. It actually produced the mRNA, and we were able to actually detect the presence of the mRNA, the transcription of this gene, which were now encoded for the nuclear RNA polymerase in the mitochondria, but we were never able to actually see the translation of that mRNA into protein. And we didn't know why, but what you are telling me is that that a very important regulation in these plastids or the mitochondria is how the mRNA is maintained and regulated, and so it's possible that actually our mRNA eventually was degraded and never expressed, or it needed to be processed and it was not processed accordingly. So I find it very interesting.
Yeah, right now, for example, for transplantomics, what we do is to take a nuclear genome, but we have to insert upstream and 5' and 3' elements from the chloroplast. Otherwise, the transcript is not stable. They have to be processed in order to be translated. So yeah, definitely we cannot just return it like that. So.
Okay, I would like to ask you a question, Patricia, about chloroplast development. How long does the process take? And also, is it known what is the photosynthetic status of all the immature stages of the chloroplast?
Okay, let me go to the first question. The first question, it's extremely fast. Plants have evolved in the fact that once it emerges from the soil, they have to be photosynthesized like that. So in many cases, the plastids have already half or a little bit way up, producing something that is called idioplasts. And in the idioplasts, they are not internal membrane, but they are a lot of the parts to production of chlorophyll that are ready. So in minutes, you can see how it starts getting green. It's within minutes. You can slow it, you can manipulate. And your second question was?
Partially answered it, whether these immature forms can conduct photosynthesis.
Well, not in mature, but not, in fact, once you start chloroplast, you accumulate, it's green, but green doesn't mean it's photosynthetic active. In fact, the young leaves are green, but they are not photosynthetic active. Once there are some photosynthesis going on, but not very efficient, because you need to really put the photosystem 1 and photosystem 2 in different places. It's actually quite a complex process, but it's really fast. It's very impressive.
So thank you again, Patricia, for a nice presentation.